I'm really excited to introduce someone that I've admired for a long time, Marty Morell. She has really another person who's invested her life in studying epilepsy and severe epilepsies, and she's been working for the last, at least 2 decades on neurostimulation in the brain, especially the RNS, and so she's going to be talking about the clinical trial that's going on in LGS with RNS right now, and I can't wait to hear her updates. Thank you, Marty. Welcome. You can use that one.
I'm here today to talk about a study that the company NeuroPace, which is the company that's developed a device to treat epilepsy, responsive neurostimulator, is conducting. And Randall Kaye talked just a moment ago about a new attitude at FDA and wanting to help rather than block. And so we work with the device side of the FDA. There's the drug and there's the device. And I'll tell you that the same spirit of wanting to be a partner towards win has happened in the device group as well, which was not the case in 2004 when we started our studies in focal onset epilepsy, but has been the case with this. And I also want to say that our tax dollars through President Obama's BRAIN initiative and NINDS has provided almost $10 million to do this study. And for a small company like NeuroPace, it otherwise would not have been possible. So we are doing a study in Lennox-Gastaut syndrome that is done, of course, to understand if we can treat Lennox-Gastaut syndrome with this therapy, but also to learn so that we know how to do it best. And so I am a clinical professor at Stanford. I have an active clinical practice, and I'm chief medical officer at NeuroPace as well. And I'll begin by saying that the RNS system is approved for treatment of adults with focal epilepsy, focal seizures. So other uses of it are investigational, both the trial in idiopathic generalized and this trial that we're about to speak about in Lennox-Gastaut. So you all know that Lennox-Gastaut is considered a generalized epilepsy, and seizures occur from many different places or from all over, and many patients are medically intractable and usually or rarely are candidates for any type of curative surgery. And so this is the scalp EEG, and I suspect a lot of you have seen this. So the seizures are beginning from all over when you record from here, but are they really beginning someplace in the brain? And so that's the question. Where do the seizures start? How do they spread? And could we apply an electrical intervention or interruption that would keep them from starting in the first place or keep them from spreading so that a small seizure does not become a big seizure? So the RNS system was just an idea in 1997 and was approved for focal epilepsy in 2013. And it's a personalized system that monitors the electrical activity of the brain through leads or with electrodes that are placed where the seizures originate, in the region of onset or the region of spread. And it is sensing all of the time, it's recording abnormal activity, and when it sees activity that is not normal, as determined by the physician, it provides stimulation and response. And then all of that information from the neurostimulator, which is the little computer chip you see to your left, it goes to a secure website or database where all of the information on everybody is stored, so that if I'm caring for your child, if I want to see everything this device has collected over the years, it's there for me. And I'll show you why that might be important. So this is a device that's entirely contained in the head. That computer chip with the battery is placed within the skull. It doesn't go below or above the skull, and it curves with the skull. And then the leads are placed by a functional neurosurgeon with stereotactic guidance in the OR at the region or the focus that we have identified. The family goes home with a remote monitor. They can pull information from the neurostimulator and send that to me, to the physician, over the internet to the patient data management system. And in the clinic, I use a wand similar to VNS and also retrieve information and also then make changes based on what I see what I hear mostly from the caregiver, and what I see from the electrical activity that's been recorded. So this is the basic concept. It's like a cardiac defibrillator. Most of the time, everything is okay. And so the device is just monitoring, but when unusual activity is detected, here in blue, a brief pulse of stimulation, which is not perceived, is delivered, and then the device looks to see if things have normalized. Now, I mentioned that it's approved in focal epilepsy, and there's over 6,000 people, adults, who have been treated, and over 500 of those in studies. And the blue that you see is what FDA asked us. They asked us to show everywhere that a lead, an RNS system lead, had been placed in the clinical trials. And so you can see there's experience lots of different places. And we know that for focal epilepsy, it works. Now, the blue that you see is the median % reduction in focal epilepsy from year 1 to year 9, and you see that it's getting better over time. And then if you look at the orange, this is the contemporary experience. So that orange shows that the response is better, and it continues to improve. And what's the difference between the blue and the orange? It's that we've learned all of this interaction with families, with the individuals who are participating in the trial, and by looking at the data, continues to teach us how to do this better. And how are we learning? Well, on the top, you'll see an intracranial EEG, and this is EEG activity collected directly from the brain. And where you see the blue is where the device has detected that a seizure is occurring. And it is then telling me how long it lasted. And on the bottom, you'll see this chart shows from May until January, so half of a year. And the green is every time the device has made a detection of something that's abnormal. So I can look at this and say, all right, so when we started out, things weren't so good, and then we've made changes maybe to the RNS system and how it's providing the kind of stimulation I'm providing, or maybe it's the introduction of a new medication, or maybe it's a change in sleep, or behavior, or exercise. So why responsive stimulation in Lennox-Gastaut? Well, the thought is that if this is not truly a generalized onset, if it's actually starting somewhere, then we could individualize it to each person's focus. And we know that we can detect the onset of abnormal activity within 100 to 200 milliseconds, and we can do that anywhere. And we know that we see cycles. There's daily cycles. You guys know this. There are times of day when your loved one is more likely to have a seizure, and other times of day when they are not. But we've also learned that there's periods over a month, or over 48 days or 72 days where there are cycles as well. And we can see that with this device. If one can deliver it responsively, then you don't need to provide stimulation all the time, so there's less. And then the data that I showed you can help me understand other treatment decisions. So has this been used? Well, FDA refers to this as according to the practice of medicine. And your healthcare provider can decide or recommend to you that your loved one receive any treatment that in their medical estimation could be of benefit and would be acceptably safe. And so although this is not indicated for use, there are publications. This was just one of the first ones. There are a number now. And so when we saw this, this was very informative to us. It was 2 pediatric patients who had drug-resistant epilepsy, LGS, and autism. And what you'll see there is an X-ray, and I'll just refer you to the image on the right. This is this MRI they're looking at you. It's actually an X-ray. And you'll see that the lead is placed deep in the brain. And so that's at the thalamus, the central median nucleus of the thalamus. And down below, we see the recordings. So we saw from this that we could record these seizures deep in the brain, and that we could see that lots of time there was no seizure happening, but that when there was something, that we could detect it very quickly and we could provide stimulation. And then you'll hear later about one of the individuals fundamental to this, an incredible epilepsy group at University of Melbourne in Australia, conducted a trial of deep brain stimulation of the central median nucleus of the thalamus in patients with LGS. This was a small prospective double-blind, meaning half of the patients were randomized to receive active stimulation and half were not. Everybody was implanted with the device. This was a device made by Medtronic. There were 3 months of active versus sham, and there was a reduction in patient-reported seizures. So we went to NIH and we said we really need to know what the networks are for seizures. And we need to know whether stimulation is going to work, and we need to understand how to provide the stimulation, how much current, what frequency, how long the stimulation pulses should be. And so they provided this funding for a prospective study, a crossover study of Lennox-Gastaut. And we wanted to know, is this starting in the frontal lobe or is this starting in the thalamic nucleus? Because the University of Melbourne's group suggested that it might lead in from the frontal lobe. Now, our device can be connected to 2 leads. So in the individuals participating in this study, there are actually 2 neurostimulators placed so that we can have 2 leads on each side. There are 20 patients who will be participating, and we need to know how best to do this. So there are 3 different conditions. One condition is very fast stimulation that's really short. Another is lower frequency stimulation that's longer. And the third is no stimulation at all. And the patients and caregivers do not know which arm the individual is in, and then it's an open label. Now, one thing you'll notice, 12 months, device trials are really long. It's just the way it is. So in pharma trials, it may be 12 or 14 weeks, but in device trials, it's a year, and we're following individuals out for 2 years. So talk about diary fatigue. We're asking caregivers to document every time there is a drop seizure with a fall or a drop seizure that would have led to a fall. And so one thing we've learned is that maybe this is too burdensome and we need to look at other outcome measures. So I've been very interested in hearing about all the work in how we should measure success. FDA said you can, FDA said enroll 12 and up. NIH said we're a little nervous about that, do 15 years of age and up for the first group, and then 12. We want to see the safety, and the safety was good in the first 10, so it's now 12. And it's a lot of seizures, but for you guys, many of you will say 5 drop seizures a month, that's nothing. But that was the criteria. And the diagnosis of Lennox-Gastaut had to be established, or has to be established, by the scalp EEG that you have all had with the characteristic features. What are the goals of this? Well, one, I said it's safety and effectiveness. But the other is we want to know where to put the leads, and we want to know that before we put the leads in. So can we define non-invasively the best place to put the leads using neuroimaging? And I'll show you that information. And then afterwards, once it's placed, can we use all of the electrical data we're getting so that we can very rapidly figure out the best stimulation settings for that individual and not wait for you to come back every month or 2 months or 3 months and tell me how your child is doing? I would love to know by looking at the data very quickly how your child is doing. And then, of course, we want to use this information to continue to develop a better neurostimulator. Now, the first patients were enrolled the end of 2022, and this is that pre-implant information that I was talking about. So this is really beautiful. It's using neuroimaging and computer analysis of that neuroimaging. And what you see upper left is the thalamus, and we can map out exactly within the thalamus and all of its nuclei the target, which is the central median nucleus, which is a very small target. We can then identify the trajectory the surgeon should use so that the lead, which you see illustrated down below that with the electrode contacts, so that we can make sure there are electrode contacts in that target, which there is pink. And then we can look at the MRI to look at the connections from the central median nucleus, which is the colorful little blob you see there, the connections that go to the cortex or to the surface of the brain, so that we can put an electrode in the right nucleus in the thalamus, and then by looking at these connections, we can place the lead over the frontal lobe where that is most connected in that individual. And surprise, not everybody is the same. So here's an example. We start with a regular MRI scan. There's then a computer analysis that's done. Just follow the arrows. We then look, and you'll see in the red where the greatest connection is with that nucleus in the thalamus. And then preoperatively, the neurosurgeons meet with the epileptologist, and there is a plan about where to put the lead on the surface of the brain and where to put the lead in the thalamus. So I can't tell you the results of the study because it's underway. We have 19 individuals of 20 who are enrolled. But what I want to show you is this, because here's the first answer. The first question is, where are these starting? And you heard earlier, they're not starting all over at the same time. And the top, you'll see electrodes that are in the frontal lobe on the surface, and the bottom two channels there are from the central median of the nucleus. And where you see the red is when the activity in the brain is picking up and becoming epileptic. And so we can see here that we can detect it right away. I call it the Nike swoosh. And that is when the brain activity is switching from normal to abnormal. And then we see the spread to the central median nucleus. Now, this is the kind of information that helps us understand where should we be treating in the brain? Where should the target be? And how should we be treating? And so I do want to say, and I think you've heard this, and I heard about Dr. Worrell's wonderful slide yesterday of peaches and other orange fruits. And there are a lot of people who are like each other, but we've learned, any of us who have been dealing with loved ones with these disorders, and any healthcare provider who's been treating them, and I have for a very long time, know that although there are some that are similar, there's nobody who's exactly the same. And so it would be naive to say that there's going to be one therapy that's going to knock it out of the park for everyone. But I do believe that if we keep working, there will be a therapy that works really well for each person. But what our goal is, and what our responsibility is to be able to meet with you and your child and say, based on all of this information that we can put together from pictures of the brain, from recordings, from what we know from the science, this is the first place we should go. And so that's the challenge, and that's our responsibility. And I just want to tell you that the world is a bad place, and it's ugly, and we can get upset about everything that's going on. But I see patients all day Monday, and I come home on Monday, and my family always says, those are the days that you are happiest. And I say, that's because these are the days when I see how spectacular people are coming together. And so that's you guys. These are the centers that are participating, and this is a tough study. The families, 2 years of keeping seizure diaries and all of this stuff. So we do have a contact information for our pediatric nurse practitioner who works with me in medical affairs for any questions. Thank you.
So we have time for one question. Anybody have one burning question right there? Oh, we got our mic runner right there. Thanks for the volunteering. LGS mom by day, mic runner also by day.
Are there, I see the RNS is trademarked, so is it only manufactured by NeuroPace?
So there are 3 neuromodulation devices approved by FDA, the VNS. You all know about the VNS. The deep brain stimulator, and those studies were done by the company Medtronic, and that provides, similar to VNS, a certain amount of stimulation. So generally it's 30 seconds of stimulation in the thalamus, and then 5 minutes off. So it's 30 seconds on, 5 minutes off. It does not collect the information, and it doesn't respond. So the RNS system that was developed and is manufactured by NeuroPace is the only device that is responsive.
Great, thank you. I know that Marty is going to be here if you have any questions, and I know there will be a lot. Thank you, that was great. And